So welcome everyone, and this is my pleasure to give the 8th webinar in the series of Polonium Foundation webinars. It's also my first talk of this kind, so it's really interesting for me. Hopefully we will have some fun here. As mentioned, my name is Marta Gajowa, and I'm currently a postdoctoral fellow in UC Berkeley in California in the group of Hillel Adesnik. And today I will tell you a bit about my scientific project. I'm working in the lab and some background of it. And the project is focused on one of the crucial components of the brain circuit organization, which are synaptic connections, but in a new way because of new technologies which were developed and which were developed relatively recently. So I will start my talk with to give some focus on the context, to give you some idea about what are the biological questions I'm looking to answer for. And next I will describe technological approach which allows us to revisit some topics and basically ask the same question as were asked already 50 years ago, but with a chance to give some new information and new insights. And at the end, I will show you some very first results, basically a promise of a results, more or like a proof of principle that what we are doing is possible. So let's start. So as a neuroscientist, I'm interested in how does the brain work? And what I understand for this general question, of course, is influenced by the field I'm working in and a specific structure I'm working with. So let's start from stating that at the beginning that I'm working in primary visual cortex of a mouse. This is like in the brain, if you have this cartoon of a mouse brain, visual cortex, primary visual cortex would be this purple area and like extended a bit here. And abbreviation is V1. I will be, whenever I talk V1, I mean primary visual cortex. And if you would slice through such a brain tissue, piece of such a tissue, you can have an image like this one on the right, or the left, I don't know. This is how the neurons looks in a big magnification. I'm giving you today a bit of a circuit perspective. So basically I'm interested how does it work that when we see something through our eyes, this goes on the way through a few structures in our brain, and then the primary visual cortex is first placed in the cortex when the processing of this signal occurs. And as you can see, around visual cortex, you have some secondary areas which also process the visual signal, but the primary visual cortex, as it first, it's kind of a starting hub for all of this information which is there, which is why it's important and why it's interesting to study this particular area in order to understand how the vision works and how any kind of processing in the brain works. So, okay, so there is like an old sentence, which is kind of a neuroscience dogma in the field: what fires together wires together, and every student of neuroscience or even biology learns that sentence at some point. And this is a really easy sentence and nice to remember. It stresses the fact that neurons needs to be connected to each other to pass the information. It was also used in a context that, the used synapses, the used connections are getting more and more strong over time. But in our talk, we are not focusing about the time influence this time. We want to understand really quickly in a time we have now, like if you see at a given moment an object, how does it work that you see it? What kind of processes occurs? So we are totally detached from like some longer perspective of time here. We want to understand a given snapshot, a given moment. And as you can see, the photo I'm presenting here comes from my recordings, and it's kind of similar to these two red neurons, which on the cartoon on the right, which shows you a presynaptic neuron and a postsynaptic neuron. So the first one, the presynaptic, is someone who gives the information to the next neuron, which is a postsynaptic neuron. And now the point of connection between them is called synapses. So coming back to this Donald Hebb sentence, what fires together, wires together, this sentence for a long time was a kind of focused the attention of a scientist on anatomical studies. So on the picture on the right, you have two neurons which are perfused with red dye, which enables you to see somehow the structure of these two neurons. And you can see that they're a bit like a trees, like they have some branches, they overlap a bit, but you quite really don't know if they are in contact or if they aren't. And of course, this is just a picture from 2-photon microscope, really simple. But over years, scientists were trying to take really detailed pictures, which will show you the place of contact between cells. So they could study them and recreate what could be the function, what could be the role of synapses, or what we could learn more. But there's one big bias of this kind of studies, of anatomical studies, that you don't have any idea if the connection is used. We just know that it is there. So what we can learn from photo is like we can learn from the map that, well, there are paths, and there's several of them, and there's then several possible ways we can take. So for example, if I want to go from my home to my work in the university, I can take, well, Google shows me at least 3 routes, which he found that could be useful for me. And which one I take really depends on the circumstances and on a given day. So sometimes I'm in a rush. Sometimes I'm look where's the lowest traffic. Sometimes I won't go on by foot, so I would like to go next to the park. And that is basically our circumstances for me, which helps me to take the decision which path I'm choosing. And nowadays, like map gives you a lot of contextual information, which helps you to take these decisions. So how this can we think about this in the context of neurons? What could be the circumstances for neurons? Because already from the photo we learned that there's a lot of branches and a lot of possible paths. So which of the paths the cell will take when visual stimuli is processed? So some circumstances of neurons, I will give you 2 examples, the 2 one I'm really interested in my studies. So first is a function of a cell. So we would like to study the circuit with the information of the anatomy of the circuit, but also of the function of each cell in this picture. So the piece of information you need to know at that point is that neurons in primary visual cortex are tuned to various features of presented image. So for example, if there is an object in your field of view which moves vertically, some neurons will notice that stimuli and will fire and will tell other neurons there is this kind of stimulus around. On the other way, other neurons will fire if the stimuli is moving in another direction. And all the word we look for can be kind of simplified as really simple objects which moves or are stationary, and they have different contrasts. So this is why, like, scientists study this kind of visual responses with really simple stimuli, which is like moving grating, for example. And I will show you an example of a movie. I hope this can go smoothly, of how this could look like. So this will be a movie which I recorded during my PhD, and this is coming from extracellular recordings in primary visual cortex of a mouse who is anesthetized, but viewing the grating of on the screen, which you will see that they are moving in different directions, and you will be able to see the spikes here on this small screen, which is the oscilloscope, and hear them actually. The noise, it's like sights in action. And I
don't think we can actually see it.
Okay,
I can hear it, but not see it.
Should we start to do new share?
Yeah, that might work.
Yeah, I will share this. Do you see it now?
Yep.
Okay, so you can see some spikes. Can you? Yeah. So this is like the neurons in action, a single neuron action. this is the stimuli we apply, and the mouse is anesthetized here, and there was some electrode inside. And these are the responses of the same neuron for different direction of the stimuli. In each of these boxes is kind of another direction. And when we analyze that, you see that there's a different amounts of spikes. The bars tells you amounts of spikes in time. And this has some nice structure. And this is the kind of thing which we are looking for in our studies. So let's come back to the slides. Okay, we're back. Okay, so as you could see it kind of in the movie, there is also like my current boss, my current supervisor works on this topic too, and he published a really elegant paper when he did kind of similar studies, just using patch clamp in vivo, and he would manipulate not only the direction but also the contrast of the stimuli, and you can see the proof that neurons indeed are tuned for some visual features. You can see that there's more spikes, more of these like 0 and 1 information. Neurons communicate to each other with the help of the spikes. So this is one contextual information I want to put my study in. And another type of it is that, well, there's actually many neuronal types. And the picture I have here just shows you 4 different groups. There would be like pyramidal neurons, which are excitatory neurons. There would be PV, VIP, and SST neurons, which are inhibitory neurons. And they talk to each other in a different way. And this kind of image is recently accepted in the field that, well, more or less the connections occurs this way, and functionally this would have sense that the circuit, that certain types of cells talks in a given manner to the pyramidal cells and vice versa. But this is still something which is under investigation, including in my work. And if you would like to compare this to something more embedded in the field of neuroscience, this would be like Lego bricks. So you have different colors of Lego bricks, different types of them, and can build several things. Just you need to do more and more of the same type of Lego bricks. So yeah, so now how do we build a map, and what is really the purpose more generally? Now, okay, we have this. So I also want to state at the beginning that the biological motivation of my work is not only my work. There is a few other labs and a lot of other researchers who are interested in similar topics. They work with a bit different techniques, so it's not like super unique. But the principle of biological question is like, I would like to have the conversion from photo of a tissue or living tissue in a living mouse to a map transformation of connections. And I would like to have this map and in a high throughput way. So like I could obtain this pretty fast and at a given moment. And what I understand by the map, it's a descriptions of how the cells are connected to each other and how strong are these connections. So give you a bit more example, if you would zoom in this picture of the two fields with the dye cells, I would like to do on them an experiment which will enable me to stimulate the first cell, which will be a presynaptic candidate, and I will read the response from the second cell, which, if there is a connection, I will be able to record it. If there's no connection, there will be no difference in what I am recording. And this is somehow also traditional way how these kind of studies were done in the past. So you see these two kind of triangles which basically mimics electrodes. As you can see on this photo here, there's like a red triangle, which is a small glass electrode. And in principle, how you would do that traditionally, you would put 2 small electrodes, one on each neuron, and then stimulate with 1 electrode, inject some stimulation to 1 neuron, and wait if the second is responding. And you can imagine that putting these electrodes is kind of hard for such small neurons, you could see like a scale bar here is 15 microns, and this is more or less the size of the soma, and the needle of the tip has 2 microns, which is really, really small. And what was established before, a probability of connection between pyramidal neurons is about 10%. And we don't know anything yet about what's the functional context. Well, there are some presumptions, but it's really local, and it's really tedious work to learn what the connections present or not. And if this is 10%, so you can imagine that then you need to test 100 pairs at least to find 10 connections, and not every approach will be successful. And this gives you the idea how tedious work this can be. This can take years, and no one has years nowadays in science. So the idea is to replace some of this tedious work and do not use the electrode here anymore, but only use one electrode on my postsynaptic cell, and try to find a way to test cells around, all the cells around, which may give you the connection. So how we do that? Let's move to the technical approach for a bit. So we are going to combine 3 techniques. One of them is whole cell patch clamp, which you already had some intuition about. This technique is a gold standard. It's about 60, 70 years old now, and it gives you access to the information the neuron has. And I will speak about it more in detail in a while, but basically what we are recording is the neuronal information. So the second technique we will use is optogenetics. It's about 20 years technique. And basically this technique allows us to make neurons light sensitive. So we put an ion channel inside the neurons. I will come back to it. And the neurons who has these ion channels, we are able to trigger them by light. At the end, we use 2-photon 3D shot holography, which is a technique which enables us to shape the light in a shape of cells. So it provides 3D pattern of blobs of light, which are more or less of the size of the soma, and as you already intuitively can probably infer, the optogenetics and holography gives us ability to stimulate cells around our postsynaptic neuron. So let's jump a bit more into it, so it will be probably a bit more easier to follow. The whole patch clamp technique. So I was referring to this technique as putting a needle on top of a neuron. And it's kind of really abstract, but with this photo, I hope you will have more perspective and you will be able to imagine it easier how this is done. So this is the front view of the setup I'm currently working. There's an objective here. There's a small chamber here in which I put a tiny bit of tissue, and there is the electrode here. So you see that basically I'm not putting it manually. There's wires here, there's joystick to use all of this, and I can approach a chosen cell on my screen, which I see, and I can touch delicately this cell. And when I will touch it, well, good enough, in a way that it's really tight between the membrane and the glass capillary, I can apply a small suction to this cell, which will basically open the piece of the membrane under the glass capillary. And this way, the inside of the cell and the inside of my electrode, which is filled with some special solution connected through the silver wire to the whole amplifier and a lot of equipment, enables me to put the cell and my electrode in one electrical circuit. And because of that, I gain access to what is going on in the neurons. So to give you a bit more intuition, to not talk only about electricity, because not everyone is able to easily follow that, imagine you have a balloon filled with water. And now you take a needle and you want to delicately pierce the balloon. So the balloon is with water, you have this needle, so if you will put it inside, there will be some pressure, like just before you pierce, yes? there will be like a kind of a outside layer of balloon pressure, which will be different. The strength of this pressure, of the resistance for the needle, will be different depending how much water is inside the balloon. So this you could compare to like potential of a neuron. Like neurons ourselves, we have a difference of potentials between inside and outside. And if you would do a small hole with this needle, some water would leak out. And how fast this water would leak out depends on how big is the hole you did. And this you could compare to the current flow between the inside and outside of the neuronal membrane. And indeed, neurons have actually small holes in the membrane, which are ion channels, which basically maintain the potential in between neurons, and neurons operates by the difference of the ion channels composition inside and outside. And this can be recorded with whole cell patch clamp technique. When we have the access, it's single electrical circuit, and we can see the membrane potential changes, including the spikes, which can be compared like 0 and 1 code of the neurons, and the current, which is sometimes bigger, sometimes smaller, but we are also learning some information from that. So that's basically the neurons juice, the current and the potential. That's the information we're learning from neurons. And this is some kind of neuronal language. We're trying to learn what is this language and what's the grammar of it. So jumping to the optogenetics, another tool, as I told you, it's an ion channel. It's an ion channel which was discovered at the beginning of the century, that it is in the alga. It helps alga with the phototaxi. And some scientists have a brilliant idea to put this ion channel in the membrane of the neurons. And then they started being able to trigger the neurons by light. So when you shine the light of such ion channels, they open, they enable some positive ion charge to go inside the neurons and basically mimic what physiologically occurs during normal neuronal operation. Just we can do it on demand. And it was really big, discovery at the beginning, and it quickly rise to an entire subfield in neuroscience. And there is many of different types of neurons, of opsins, which enables us to study neuronal circuits. But through about 10 years later, another type of opsin appeared, which is the opsin I'm working with. It's the opsin which was engineered in the lab I'm working with by a really talented person. And what's particular about this opsin, it enables you to trigger single spike in response to light pulse. So to explain what we have of the picture, I was doing some experiments with this opsin when I was first stimulating the neuron with electric stimulation, giving some current step of electricity to the neuron. So the neuron would respond with a first action potential. And then I was giving different numbers of light pulses. And as you can see, you have a nice response of 1 to 1, 1 pulse for 1 spike response in this picture. And was also published by the lab in the paper, showing that there is the fidelity index of spiking almost 1. So it's really rarely fails, and really rarely gives you more than 1 spike. And this tool finally gives us enough time precision to study neuronal circuits in detail. Yes, optogenetics was great so far, but it only allows us to trigger the cells with more or less precise manner. Finally, we have the tool which gives us the precision of deciding how many spikes we want to put into the circuit, which is much more precise. So this is the option I'll be using. And that's another movie I would like to show you, just to give you some more intuition how this works, how opsins works in reality. So there will be an opsin expressed in CHO cells. Not this. Do you see that picture now? Okay. These cells are not neurons. These are Chinese hamster ovary cells, which are basically cells which almost do not have their own ion channels. So researchers really often use them to put some ion channels there and test the properties of these channels. And this is what exactly I was recording. And this is just fun to watch. There will be some light stimulation put on this blob, on this cell, which I patched again. And then you will be able to see the deflection from this line here, which basically tells you how much current was evoked. So the nose is the shutter of the laser. You can see some deflections already. And you can see that the deflection is changing because I'm changing the time duration of the stimulation during the protocol. Okay, I think everyone could see already some deflections in the membrane. Okay. I think I need to share again. I'm back. Is it okay? Okay. And the third technique, which is two-photon 3D shot holography, which is a type of holography. It's also a relatively new technique. It started being, like people start to work on this kind of setups 10, 15, 20 years ago when the field of optical tweezers started to being a thing in the science. And basically then with the progress of optogenetics and having the ability of trigger neurons by light, we needed better microscopes to be able to stimulate the neurons. So that's the schema from the paper. Of course, everyone is welcome to read the paper, but probably it won't tell you a lot unless you are not an optician. This is though how it looks like in practice. So you see that there's a bunch of lenses here basically and optical elements, and they do something to the light beam which goes out from the laser. But there are really two important elements here. First one is the diffraction grating, which is here on the photo, which is basically a type of grated mirror which makes a rainbow from a laser beam which is incoming on that. And by this, it's like splitting one color laser to different colors, which is different wavelengths. And then at the end, you can see you have like a rainbow here matched. And then on the moment when we need to, like, when the light comes out from the objective, by all the other lenses and the other elements, the light is put back in focal plane at the same time, but in a really condensed manner. Basically, the whole point of making a rainbow is to split to different light components, which will have different delay, and they will be differently delayed over the other components of the optical elements. And when they will be put back together, you get one really strong pulse of really a lot of photons. Because what then triggers our ion channel in the cells are the photons of light. And we want them to be, we want to maximize the amount of the photons which touches our ion channel. So that's one important element, and this element enables us to have really nice precision in vertical, in up-down Z dimension. The other important element is SLM, which is spatial light modulator, which is a kind of a screen with pixels which change the phase of the light, which enable us to give the XY dimension of our dots, of our blobs of light. So this is like how you could shape this elephant you could see before. And the SLM is basically here on our setup. So it was definitely a fun of building it. We did not invent that, it was invented before, and a bit different configurations also in different labs. But definitely building such a setup was a fun, and building like Lego technique kind of play. And to show you some kind of principle how this setup could work, there is these two pictures. So basically, this is our everyday operation. When we come and do experiment on this setup, we need to check if the optical alignment of the setup is still good enough to do our experiments, which basically means that we put a fluorescent light below our objective and we try to burn some shape on this light with our laser. Here we try to burn like this kind of eagle, which the wings of the eagle are in different z dimensions, but the middle of it is perfectly in zero plane. And this is our burn holes picture of exactly this type of the shape, when you can see that on the middle you have perfectly in the same places, but it's only in the middle which shows you that it's good enough for this day. Your zero plane is where you want it to have, and if you will put a cell in place on one of these dots and the cell will have opsin, the cell will respond. So to close this technological part now, I would like to summarize a bit of it. So we learned that what more or less is whole cell patch clamp, and we learned that it enables us to learn the information between the neurons. In the context of mapping project, it will enable us to detect the presence of synaptic connection and its characteristic. So we don't have anymore an electrode here, we have electrode only on postsynapse, like on this picture, and we will be stimulating around all the cells with our 3D shot holography, and the cells which will have opsin will trigger a spike. And if the cells are connected, the one which trigger a spike and my cells under the electrode, I will see that in the presence of a deflection, and this will be my signature of synaptic connection. So this way we achieve this high throughput way of mapping. As well, light is much faster to move a light block from place to place instead of moving the electrode, yes? So this enables us to, within a few minutes, have the entire map of connections from a single neuron. So now I would like to show you a bit more of our protocol, how we do it really, and how we obtain this photo to map transformation. So we have our cell filled with a green dye this time in a slice of the tissue, and we will shoot holograms. We'll shoot blobs of light in all possible places in a kind of rectangular manner in several planes above and below our cell, and in all places around, kind of a cube way. And if the cell is connected, we will detect with electrophysiology, and then during the analysis we'll translating the amplitude of our synaptic connections to basically a pixel of color. And the hottest, the more bright the pixel, the bigger was the amplitude of the connection, the stronger was the connection we recorded. So this way we also obtain a photo of our tissues. We do a really precise scan of our tissue after all. And so we will learn the anatomical structure of the piece of tissue, and we will know the connections between it. And that's the in vitro part. In future, the idea is to go towards in vivo studies and to do it as you saw more or less on one of the movies before, and put some grating to the mouse and kind of obtain the mouse when the mouse is viewing a stimuli. And that in vivo step will enable us to learn this functional context. So a bit more words for the ones who were really interested and really were able to follow so far. This is the cube of the holograms we do. We apply, we calculate, and our cube dimensions are more or less 160 by 160 by 153 microns so far. And we do different kinds of this. Which is one of example. There is some time duration between the stimulation in this place versus in this place. Let's say the time also matters to be able to really record from where our deflection of current is coming from, yes? Yeah, okay, so this is basically how it looks like when we are doing the experiment. So this is some example of raw traces in the plot. So traditional, the top one is a bit zoom out perspective, that the bottom one is zoom in of this part. And what you can see here is basically evoked photocurrent, which is basically, so whenever we stimulate the cell with opsin, with our one of the holograms, the cell is triggering a spike. And if the cells are connected, you get this kind of deflections. So you can see that there's quite a lot of them. They are not super big in relation to other synaptic connections. But you can see that there's a lot of these places when you have this deflection, which is basically the current coming to the postsynaptic side of the cell. And the gray bars here basically is plotted our time when the laser is on a particular position in our grid, and we repeat it about 10 times in each conditions. And what's also important, we are randomly shuffling the position. So each time the cube and the order we stimulate our cells is different, and it just helps with getting the more real picture. So this is how it looks in raw traces. And this is how looks the photo of such a cell when we have the white circles here are the one plane of the grid we put in the XY dimension, just so you could appreciate more or less, like there's a field cell with some branches here, and there are cells around the green one. And the very first step also to learn if there is a sense of recording from this neuron, we are doing one photon LED stimulation, which is basically a colorful bulb, a bit more powerful than the normal bulb maybe. And at that point of time, when we flash one photon light, all the cells which contain opsin spikes, and all of them at the same time give us the synaptic input to our cell under the electrode, which gives you this really big, massive response. And this basically tells us, yeah, this cell has a lot of connections and we need to find them. And now we would use 2 photons to have this kind of raw traces we had just before. And also a really important thing here to stress, how do we know it's synaptic? How do we know it's not from the cell we are recording from? We learn it because of the synaptic delay between the onset of the stimulation and the start of our stimuli. So the time, there's still some time, the presynaptic cells needs to spike, and the stimulation needs to go through the synapse, and we need to have, there's some delay time to be able to record that synaptic input on the cell of another postsynaptic cell. So this approximately gives you 4 milliseconds. This is how we learn that this is synaptic, that there's a lot of inputs. We want to find where are the connections. So after we put our cube, we basically obtain a pixelized map or where were the connections. And here I plotted some of the points from this pixelized grid. And you can see the really yellow pixel A, which is here, which is giving me each time almost when I stimulate a presynaptic cell at that point, my cell, which is more or less here at in the case of this cell, is giving me the response. And you see that here there is only like blue light, and there is no really a response. Like the yellow pixel, like yellow-green pixels, show you how much current was evoked on the different repetitions. So the overview of whole experiment basically now is like we have this cell with the pipette. This is our postsynaptic cell, which do not have opsin. And we have a map of connections at different planes, like above the cell, at the cell plane, and a few layers before. Where are the connections this cell obtained? And with this blue light, I put some positions which were inferred from the maps where the connections should be. And this is not the best photo to see that, but at least some of them will overlap with kind of the photo of the true connections. And now the next step will be to take a nice elegant stack and analyze this for each plane. But this will be kind of the future work. It's still the work of progress, and this is like some maps, some 4 examples of maps which we collected recently with this protocol to see how diverse is that. You see, like there are different cells, they have different morphology, the kind of the branches of the neurons are different, and also the places, the hotspots are a bit different. And now the next step will be to really analyze what are the rules, how far are the connections, how strong are they, and put it in more and more context, and this will give us the map at the end. So that's the end, and I would like to acknowledge a lot of people. So first of all, I would like to acknowledge my supervisor, Hillel Adesnik. It's a great person from whom I'm learning a lot. And then my 2 rig mates, Masato Shadahiro and Udaya Yagadishan. We were working together before pandemic. We started building this, the microscope. It's a photo when we basically finished and we're doing a bit of celebration. And then with Masato, well, the pandemic time came, but after 4 months of staying at home, we were allowed finally to come back to the lab, but we needed to work with a special gowns and special face shields. And well, I'm happy it's not really the situation anymore, but it was fun. And I would also like to acknowledge the whole Adesnik lab. There's a group of really talented and smart people and really nice people who are collaborative. And this project is funded by the NIH U19 funding. And I would like also to thank you, the Polonium Foundation, for organizing the webinars and giving me opportunity to share my work and well, maybe have some fun even. Thank you.
Thank you, Marta, for a great talk. And it's great to see that you were also able to continue working even during the pandemic. Do we have any questions? We had one question in the chat, which came up while you were talking, I think, about the patch clamp technique.
Mm-hmm.
And it says, is it in vitro?
Yeah, so far the project is in vitro. Although I spent some time under my PhD on patch clamp in vivo, so that's my natural next step to move it pretty quickly to in vivo.
Right, okay. We have one person who wants to ask a question. I will unmute this person. Hello?
Hello, do you have my sound?
Yes, hi, you can ask your question.
Hi, thank you for your great presentation. It was really interesting. So my question is that, so you can understand with your technique that there's a connection between a neuron that you're exciting and a neuron that you're recording. So my question is that, can you understand if they are connected directly or there is some intermediary neurons between them?
Yeah, so
is it clear?
Yeah. So that's actually a really good question. I was not talking about it to not maybe mix up too much. Yes, so with the technique I showed you, the whole point is only to detect monosynaptic connections. So the direct one, the one which was just one synapse before. And well, I think in the raw data, I sometimes see some signature of not monosynaptic connections, like 2 synapses or more, but it's really rare, and basically it really depends on how well you will tune the power, the use of your laser to your opsins. So which is like, you know, I just show you some proof of principle that it's kind of possible, but there was years of studying how to do that, how to choose the right laser power, what opsin to use to be able to really detect the connections in the reliable way and monosynaptic way.
Right, so you can understand that if it's direct. So So the way is by setting the intensity of your light, right?
Mm-hmm.
And the option.
Yeah.
Thank you. Very interesting. Thank you.
Sorry. We have someone who wants to ask a question. I'm guessing you want me to unmute your microphone.
Thank you.
Okay.
I think we have another question in chat, but now I can ask my question. I am working on mostly memory processes, and I would like to ask, because you are working with primary visual cortex, as I understand.
Yes.
And after you stimulate, and when you finish your stimulation, do you also get similar activities during, for example, for 20 seconds or 30 seconds, as a just some kind of short-term memory in your visual cortex?
So
Do
actually,
you remember?
yeah. So this is the thing we want to avoid. We do not want to have any kind of plasticity, yes? Because then we would strengthen some of the synapses. So the whole point of the technique is that we move the light super fast and we do not stimulate overly the same place with any given frequency, yes? So first, let's say the mapping, the visiting on each space in this cube, take us approximately 1 minute to visit all these almost 3,000 places in the cube we are using so far. And within this 1 minute, we are visiting each place only once. So the frequency of stimulation of the same cell is too low to talk about any kind of potentiation or any kind of strengthening of the synapses. And overall, the whole mapping and collecting the whole protocol takes about half an hour to an hour, depending on how big map we would like to have and to extend, which is, again, not the time course to really do study on the potentiation. And occasionally we apply some blockers in the extracellular solution too. So we kind of forbid the potentiation.
Okay, I understand. Thank you very much.
Thank you.
Okay, let me read one question from the chat. Do you only record from pyramidal cells or also from other types of cells in the visual cortex?
So the data I showed were coming from pyramidal cells, but yes, we have different mouse strains which will put the ops into different types, the genetic types of the cells, and this will enable us to build the maps for different types of neurons.
Okay. The next question someone wanted to ask in person, so let me. Can you unmute your microphone? Yes.
Hi, Marta. Hey, great talk and nice work. So you try to multiplex by using this holographic projection, but in your detection system, you still patch on a single postsynaptic neuron.
Yes.
Is there a way that you could use some voltage sensitive dyes and some green lenses so that even you can multiplex the readout? So in one shot, you get an entire layer of postsynaptic cells.
Yeah, so that's really a great idea. And although it's not my project, I know people who kind of try this approach, who would completely take out the electrode from the picture and would like to, read the activity of the cell from calcium imaging. The voltage sensitive dyes are not good enough yet to give you this level of precision to be able to read that for so long, because these studies, you can imagine that once you remove the electrode from the picture, you want to do it in vivo only during behavior. And just voltage sensitive dyes, you need to put them, they need to be taken out, and even if they are genetically encoded now, they are still they still work in progress. I know there are some people who work on perfect voltage sensitive dyes now who would in theory enable that, but this is still developed technology. And to also come back to my work, I'm not interested in that. I really would like to have the access to the current and to the potential information, to the juice of the neuron, and to really look not only at the spikes present, but also the current and on the underlying activity of the neurons, which is suprathreshold activity, yes, before the spike, what's going on then. And this is just closer to my interests. But yes, that what you asked, it's, well, I don't know if possible now, but there are definitely good tries to achieve similar thing in that way too.
Okay, thanks a lot.
Thank you.
Thanks. If anyone else wants to ask a question in person, then please use the raise hand function. We have one question in the chat. Could you repeat once again, what is the advantage of using opsin instead of, oh, sorry, it's gone. Let me find it again. What is the advantage of using opsin instead of light stimulation against light stimulation alone? I don't fully get, does that make sense?
Like if you don't have opsin in the cell, you won't stimulate the neuron, yes? Like neurons, it's not like, well, it's a brain tissue. In the brain, neurons do not respond by light naturally, physiologically. You need to put the special ion channel to make them light sensitive. In the retina, yes, it's which is just from our eye, and is actually a piece of the brain. In theory, you would be able to stimulate neuron by light, but it's on the only place, it's the retina.
Right. We have a couple minutes for a few more questions. The next one is, thanks for the interesting talk. Are you working on some models to explain the results in the direction of computational neuroscience?
So under the grant we are working on, there is well, entire lab who will be working on our results and helping us with understanding them and processing them with a bit more advanced level than just me as a biologist could do. You can imagine that, you know, collecting this data is a really full-time job for a few people, and analyzing this data, you need people who really specialize in algorithms, and there is a collaboration set for that too.
Right. Are there any final questions for the speaker? Okay, there is one. Do you expect different behavior in different parts of the brain that you are only investigating the vision, or is it just for consistency?
That's a really good question. So from the literature so far, we know that, for example, barrel cortex and visual cortex, they have some differences in the connectivity. But that's the point of this whole study, is actually to learn that, because what we know about connectivity so far was kind of done with this low throughput manner. We have a lot of great studies also with one-photon optogenetics about what to expect, how, or from uncaging, what could be the strengths of the connections. But one approach I am applying in my work is not only like looking at the literature and trying to recreate and being more precise, but look at this completely de novo. Like I would not know anything about that. And we expect that there might be some differences, but we are basically trying to learn from the scratch.
Great, thanks. Okay, let me ask this final question. Can one presynaptic neuron form several connections with a postsynaptic cell?
Can one presynaptic neuron
form several connections with, I might have read it wrong, sorry.
I think it's possible. Is that the way that, like, yes, so that depends how you would define the synaptic connection. Let's say in the way we detect it, we can't differentiate that really, because that would require some really precise voltage clamp recordings of several electrodes put on the single cell to kind of differentiate which connection is coming. Sometimes you can imagine that a cell has two synapses which are really close to each other. And I don't know if we have that precision to really do that. That's a really good question. Well, thank you very much for that. Yeah, in theory, it's possible to have several connections in between two same cells. Yeah.
Okay. I think that's all the questions that we've had for you. It's great to see that there was so much interest. Thank you so much for giving the talk and telling us about your research. And thank you to everyone for joining us, for your great questions. We'll be sending some requests for feedback, so it would be great if you could share that, share your feedback with us later, so we would really appreciate that. I hope you all have a great evening or day, depending on where you are. And I hope to see you in the next talk, which will be in 2021. Keep an eye out. Thanks.
Thank you. Have a good new year.